I'm Daniel Levine, and this is the Bio Report. HCW Biologics is developing immunotherapies to target chronic inflammation associated with aging. The company's therapeutics are designed to disrupt the link between chronic, low-grade inflammation and age-related disorders. The company believes this type of inflammation is a significant contributing factor to cancer and several chronic conditions, including cardiovascular disease, diabetes, neurodegenerative disease, and autoimmune disease. We spoke to Hing Wong, CEO of HCW Biologics, about the link between chronic inflammation and diseases of aging, the company's immunotherapies, and its platform for developing therapies that target this unwanted inflammation to treat cancer and other diseases. Hing, thanks for joining us.
Oh, thank you very much for inviting me to your show.
We're going to talk about inflammation, diseases of aging, and HCW's efforts to develop immunotherapies to address these conditions. What happens when we age in terms of our immune system, and how does this lead to chronic inflammation?
Well, I think it's very simple because think about it, when we are young and we have a very good immune system. Now, the immune system, we use it to get rid of a group of cells. We call it senescent cell. Now, they have physiological function in our body. So what happened is embryogenesis, tumorigenesis, they prevent tumor from growing. And so they have the capability to become so-called cellular senescence. What it really means is they really stop growing. And now when they stop growing, they do another function. They will secrete a pro-inflammatory cytokine, chemokine, all that kind of factor. And to do the job, basically, they will attract the other cell to repair, like wound healing, a lot that kind of function. Now, but when they finish their function, they're supposed to get eliminated by our competent immune system when we are young. So it's not a problem. But when we get old, that's the problem may come in. After they do their job, and our immune system cannot get rid of them. And so when they accumulate inside our body, and they would pump up more and more pro-inflammatory factor like the cytokine, chemokine, all combined and make our body now have this chronic inflammation. And that's very, very damaging because those factors make the normal cell and become senescent cell also. And then they make more pro-inflammatory factor, and so you have a vicious cycle here, and you accumulate a lot of inflammatory factors. Now, those senescent cells are not functional, and when they infect the other, just like virus, they infect another cell, and that cell, if senescent cell is no physiological function after all. So what it means is just destroy the tissue, the organs function, and come down to really age-related disease. So our organ just not functional anymore. That kind of reason and the inflammation tied to age-related diseases. I think it's a very good example. It's like diabetes. It's a very good example because when the beta cell, they make the very important hormone called insulin. and to adjust the glucose and metabolism. Now, when the beta cell get old, they become senescent and they no longer function. Anyway, so if you don't get rid of them, they accumulate, they make the other beta cell no longer function. So it's a cumulative effect. In the end, just the inflammation destroy the function of the cell. And that's why I think the inflammation tied to age-related disease all come down to our immune system. When we get old, we just no longer functional.
Well, I mean, go a little deeper for me. How does inflammation lead to disease states? What's the link between chronic low-grade inflammation and age-related conditions beyond diabetes, cancer, cardiovascular disease, neurodegenerative disease?
And at the same principle. And when this inflammatory factor accumulate, we call it chronic inflammation, they would make the functional cell to become senescent cell also. So, and it's not just a senescent cell to secrete those factors and make the chronic inflammation. Now those chronic inflammation come back to make the normal cell to turn them into senescent cell too. And they have a terminology there, they call it and paragram and an effect of the inflammatory factor. And what it does is they bind to the surface of normal cell, and then they turn them into non-functional cell, and just like the senescent cell. I think there's really a cycle there. Senescent cell make those factors to create chronic inflammation, and the chronic inflammation now come back and to hit the tissue, normal tissues, and then turn that into non-functional cell. Now, when they are non-functional, that means they don't do their job, and we have organ failures. Or maybe because the cell makeup that organ no longer function. And I when I mentioned it, and like the diabetes, just an example like that. But you can look at the liver function, the kidney, the same thing. The chronic inflammation basically look like very infective. They really make the normal cell become non-functional cells. And I think that's the link between the inflammation and functional and tissue.
HCW has developed a platform technology for designing and developing immunotherapeutic protein therapies. How does your TOBI platform work?
Well, the TOBI technology we created when we set up HCW Biologics. And I have been in this field for, I would say, 30 years. We built a lot of immunotherapeutics. A lot are based on the old knowledge people have based on like antibody type of scaffold, we call it, to build multifunctional molecule based on that scaffold. When I built HCW, and I know a lot of drawbacks based on that kind of scaffold. So I create this new scaffold, and we call it tissue factor scaffold. And so we can use it to decorate the molecule to be multifunctional. Whatever you can imagine, you want to decorate the molecule, you can use this technology, and then bypass the old technology, like I mentioned the antibody scaffold type of platform. And that's what we call it the TOBI technology. Anyway, so with that technology, and I have created over 30 different functional molecules. They have each one of them have multi-function, and it's designed. Whatever we want, we can just basically pop it in, look like, to make it like something in nature never happened. Now, using that and using our knowledge, the immunology, we know how to decorate a protein like this. So that kind of multifunctional protein, on one hand, can stimulate the immune system And actually, the way I say it is more look like rejuvenate our immune system. So now they can run after the senescent cell and kill them off. And because now we make the old immune system become new again. And on the other hand, I also decorate protein so you can take care of the inflammatory factors. So which neutralize them effectively. So you build this kind of platform, you just in your any imagination, what you really need to decorate them and do their work. And in our case, we build a molecule, one hand again, just rejuvenate the immune system. One after the senescent cell, kill them all. On the other hand, we're neutralizing all the SASP factor to further cut off the chronic inflammation. That's our technology, and it's a really new technology. And no one discovered that before, but we put it together now. We just put it in practice. And we have many examples, and we even publish scientific paper, peer-reviewed scientific paper to show how we put this technology together. That have been published too. And we have more publication coming out to further show what kind of molecule we create, they can do the job we want them to do.
You're looking at a common biologic mechanism across these various agents of diseasing. Are the drugs you're developing very particular to individual diseases, or do they share some kind of common approach in addressing the inflammation underlying these conditions?
Well, it's where a lot of people do where targeted. And let's say a disease target, you can actually create a molecule just target that particular disease. And in our case, it's not. In our case, it's very broad because we believe, and it's a lot of very strong scientific, credible evidence saying it, our health is really based on a very strong immune system. So when we build something like this, and we build a molecule and can really make where we strengthen our own immune system. So in a sense, and let the immune system come back to do their job, because that's why they keep us alive and keep us strong. So, and then we decorate further to really address the senescent cell. So all together, and to put it short, yes, we create a molecule, not a target in one particular disease. It's basically targeting all age-related disease because they have a common denominator. We understand if you really able to create a molecule to make our immune system strong again, you can ask the immune system to deal with age-related disease rather than one by one and to target one particular indication. That's how we do it. So to answer your question short, it is simply we create a platform and with a single molecule and can address a lot of age-related disease. And we have done a lot of work and the research and show it is true. And this molecule, the first one we create, we call it 9218, HCW 9218, and it worked really well against, for instance, cancer, diabetes, and also neurodegenerative disease. So I think it can be done, but I think the fundamental is how to create something and to get rid of what I mentioned, the senescent cell and also the factor they secrete, create the chronic inflammation. You can address a lot of disease very broadly.
Well, let's talk about HCW 9218. This is in development for a number of cancers. The lead indication is pancreatic cancer. Why start with pancreatic cancer?
Well, if you look at the molecule, we call it HCW 9218. It has two components in it. One component is we call it the IL-15. That's a cytokine can stimulate the immune system. The most powerful cytokine can stimulate so-called the NK cell, the T cell, the one after the disease cell, including cancer. Now, but also pancreatic cancer have another big problem. Why is no drug effective against them? Because the cancer cell, pancreatic cancer cell, they secrete a lot of so-called immunosuppressive cytokine, and particularly TGF-beta. This is so powerful. The TGF-beta they secrete. Their job is shut down the immune system, so the immune system cannot get to them. Even get to them, they are not non-functional because it's TGF-beta. So the molecule we create, we put a so-called trap on it. We call it TGF-beta trap. So you can emergence the drug go into the pancreatic cancer site. And on one hand, they create the immune cell, so they are very effectively with one after the cancer, pancreatic cancer cell. On the other hand, they can neutralize the TGF-beta. So the TGF-beta, the cancer cell secretes no longer can shut down the immune system. So now the molecule can one after them, and the immune system can kill off the cancer. Now, pancreatic cancer is particular. This is a major problem. And now because we have the TGF-beta trap on it, we think it's particularly designed to take care of the pancreatic cancer cell weakness, and we can get rid of them. Also, another thing I want to mention, pancreatic cancer is known. It's very fibrotic, mean all the chemotherapy, even the immunotherapy, they cannot penetrate into the pancreatic cancer because they are very fibrotic. And the fibrotic, so we call the fibrosis, is also created by TGF-beta. So this molecule, again, and the TGF-beta trap proportion do two functions. One is shut down the TGF-beta secretes by the cancer cells, so they no longer immunosuppressive. On the other hand, they shut down the TGF-beta, so they no longer can put up this so-called fibrosis and keep the drug away from them. So we hit them by many ways, and to make sure they have no defense, so the immune cell can penetrate and kill them off.
So what exactly does the drug do once it's in the body?
Well, the drug does, and simply this way, for pancreatic cancer, number one, they would secrete, they would promote and activate the immune system, particularly the NK cell, the T cell, and those are the cells capable to recognize the cancer cell and kill them all. Now, they also and would able to break up the defense in the pancreatic cancer. And so that because they suck up the TGF-beta, and so now the immune cell can penetrate inside and the activate cell can kill off the cancer. So they all happen and in the surface of the pancreatic cancer tumor, and also and inside they can penetrate and lead the immune cell to kill them all.
What are other indications are you pursuing with this?
Well, actually, this molecule could be basically enhance the chemotherapy. Now chemotherapy these days is standard of care for almost all solid tumor or hematologic tumor. But the problem of the chemotherapy is, again, they really don't kill off the cancer cell. They just really create senescent cell, so they no longer replicate. So the doctor will tell you, great, you have a response because the tumor no longer grow. But the problem is those cells would come back to replicate again. Those and then would really give you the relapse and also the so-called the metastasis to go to distant tissue and kill the patient. Now, the interesting one, I mentioned that this drug, HCW9218, it can lead the immune cell to kill off the senescent cancer cell. So you can imagine, we already have the data to show if you let the chemotherapy to hit the cancer cell, and then at the same time bring in our drug, HCW9218, you can get away the senescent cell created by the chemotherapy. In that way, you completely get away of the cancer cell. And so you can prevent the relapse and also the metastasis anyway. With that way, in a sense, you can treat all cancer and indication, not just pancreatic. You could be like colorectal cancer, breast cancer, ovarian, and prostate. This is exactly what we are doing. We are setting up another trial. Is one after the other solid tumor, and particularly focus on ovarian cancer, prostate cancer, colorectal cancer, and breast cancer. So it's really a platform and can deal with all cancer and take advantage of what the chemotherapy create for us, and using their weakness and kill off the cancer cell.
At the same time, would you expect to try it in combination with other immunotherapies?
Yeah, we do. And the standard of care today, I mean, besides chemotherapy, you heard a lot about so-called therapeutic antibodies. And they also have checkpoint inhibitors. And we have done the work to show that the power of this drug, not just enhance the chemotherapy, to get away of the so-called chemotherapy-induced senescent cell, enhance their activity. They also enhance the activity of therapeutic antibody and also enhance the checkpoint inhibitor. So the drug is ideal. That's in combination with chemotherapy, therapeutic antibody, and checkpoint inhibitor to give the patient much better response. And I think we have done that in very rigid animal model to demonstrate that. We are really excited. This is the area we get into at first.
You're also developing cell-based therapies. Your lead program here is in AML and is focused on NK cell expression. What happens to the immune system in AML, and how is it driving the disease?
Well, the disease now is, people believe again, because our immune system is malfunction when we get old, particularly in people who have the AML. So the idea to do that is really take the patient or a donor and particular focus on so-called lateral killer cell, that's an NK cell. And we can stimulate them again outside of the body, and we call it ex vivo cell-based therapy. So we create molecule, and when those immune cell, like the NK cell, when they see our molecules, and they would become so-called super killer, and we call it and so-called memory-like NK cell. And those, when you put it back into the body of the patient, and they would be able to run after the cancer cell AML here. So, and that's the basic idea. It's very, very similar to like the CAR T therapy, but the advantage doing the NK cell therapy is safety, because the CAR T have a lot of side effects, very serious side effects, and sometimes even kill the patient. But NK cell have been proven that is much safer, and they do not induce the side effect like the CAR T would and induce, and have very, very similar anti-tumor activity. So I think we use the platform technology to create molecules, particularly promote the generation or expansion of the memory like NK cell and use cell therapy and to put it back into patient the one after AML. Those are the lead product, but that's not our own focus because the company want to do simple and drug. Simply, we can do sub-Q injection, and you can do it in the doctor's office, or you can do it at home in the future, and simply just inject and then not very frequently and directly get rid of the cancer. That's the company focus. So the other cell-based therapy, we just license it to the other company, let them focus on the cell-based therapy. And in this case, we just two products. We licensed to another company. Now they are doing it. But it's not our own trial. We just license the molecule so they can use it.
Your stock is trading at a third of its 52-week high at around $2.50. How far will existing funding take you, and how do you think about funding options to advance your pipeline given where your stock price is?
Well, the lucky thing is we did a 56 million IPO, and we are very capital efficient. The money so far we are on hand and can easily last us for another 2 years without doing additional financing. And we're doing a lot of things. We really think this coming year will make our stock and much more attractive at a higher level. And that time we can consider to do additional equity financing like that. But we also, I want to mention that because we have collaboration partnership, we licensing our molecule to the other company, and we own company stock. So that could be another non-dilutive financing. We basically sell the holding on them and get the capital we need or we want to move on that without selling our own equity. So we have many different ways to do it. And in particular, when we further and further into the clinic and people knowing us outside more and more, and also understand we have a powerful platform technology and molecule can address a lot of age-related disease, I think our stock will go back up. But again, and we are very optimistic when we need the money, we should be able to get what we want. But in terms of what revenue we're going to go, either to sell the other, do non-dilutive funding, or sell our own equity, I think at that time, and we would carefully look at it. But because the long runway we have created, give us a lot of option to do so.
Hing Wong, CEO of HCW Biologics. Hing, thanks so much for your time today.
Oh, thank you very much for your invitation.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.